Hi, everyone. Welcome to our 10x-pert workshop for characterization of the tumor microenvironment with the Chromium single-cell immune profiling solution. I'm Shauna, and the 10x Community Manager and your moderator for today. Before we get started, let's go over some quick housekeeping items. Our 10 experts, who I'll introduce in a minute, will be giving a presentation followed by an open Q&A session. We're recording this session, and we'll make the recording available on the 10x community shortly. Please note that all attendees are on mute. If you'd like to ask a question, please select the Q&A option on the bottom of your screen. Selecting the Q&A option will open a Q&A box where you can type your question. Please note that you can ask your question anonymously by checking the Send Anonymously option. And finally, if you're interested in tweeting during the session, please use the hashtag 10xpert. Okay, now let's meet your 10 experts. Up first is Stéphane Boutet. Stéphane holds a PhD in biology of aging. During his university training, Stéphane studied angiogenesis during tumorigenesis and skeletal muscle regeneration. Currently, Stéphane is an application lead at 10x and spends most of his time thinking about new ways to use 10x products and combine biological data to better understand complex biology. A fun fact about Stéphane is that he did his first single-cell experiment in 2012. Joining Stéphane will be Sarah Taylor. Sarah holds a PhD in molecular biology. Since completing her PhD and postdoc, Sarah has worked extensively on the development of single-cell sequencing assays for examining the immune repertoire. Sarah now runs immunology collaborations at 10x, working with customers to explore exciting immunological questions. A fun fact about Sarah is that she developed the first commercially available single-cell repertoire profiling kit. And now I'm going to hand it over to Stéphane so he can get started with the presentation.
Thank you, Sean. So I'll start with the challenges in understanding the tumor microenvironment. I'll follow up on the factors to consider while preparing the tumor samples. And then I'll hand the cell suspension tube to Sarah, who will talk about the library preparation, show us a little bit about the data and the workflow, and with a live demo on our Loupe cell browser and Loupe DJ browser. But first, let's talk about the challenges in understanding the tumor microenvironment. So as you probably know, tumor microenvironments are very complex. And this complexity comes mostly from the heterogeneity of all the cellular components that constitute this microenvironment. So the tissue cells or normal cells, the tumor cells, and also all the immune cells infiltrating the tumor are all very heterogeneous. And probably because of that, because of this heterogeneity, it is also extremely dynamic. You can imagine that you have all the immune cells attacking the tumor cells, all the targeting the cells for destruction. We have all the tumor cells that are proliferating, and they are also dying in the process if they are not receiving enough nutrients. So we have a tumor microenvironment that has dying cells, which means there is lots of debris inside of it, and a lot of cells that are interacting with each other, which becomes a problem when we are doing a single cell experiment, because the key for a good single cell experiment is the sample quality. So sample quality is critical. We want a clean suspension with high viability. So a clean suspension with high viability means that all the challenges will be in the dissociation of the tumor. So the tumor cells are notoriously fragile, so we don't want to kill them all in the process. There's lots of cellular aggregates, there's lots of cellular debris, and when you have cells dying in this space, it means that there is lots of free-floating mRNA, which probably will bring a lot of noise in our data. So we have to be really careful in terms of pipetting techniques, all the medium that you are using, the buffer that we are using, and also the time that we spend preparing those tumors. And it can take a lot of time. So the common tumor dissociation workflow, starting from the tissue itself, the tumor, we start with the dissociation. which means either enzymatic, chemical, or mechanical dissociation. It can be also a combination of those three or two of them, to give us a cell suspension that is quite dirty, with lots of cell debris, lots of dead cells, and clumps of cells. So we can filter using cell strainers to remove the large debris on large cell clumps. And then we can, if we want to further clean up this cell suspension, we can use enrichment methods, such as FAG sorting, to remove small debris and dead cells. We can use also red blood cell lysis solution, dead cell removal kit to remove most of the dead cells. And also, if we are interested in specific subpopulation in the tumor, we can use CD3, CD19, CD45 positive enrichment kits. So I can show you now some example of what we've done in the lab with two examples of tumors. So clear cell renal carcinoma and colorectal cancer. As you can see on top, it's so those tumors were dissociated and then frozen as just a dissociated tumor. After thawing, without doing anything, that's what we obtain on top. And you see that it looks pretty bad. We have below 30% of the cells that are live. There's lots of debris, lots of clumps. So we can't go through the single cell experiment with such a bad cell suspension. So to improve that, we use filtering to remove the big cell debris and the big cell clumps. And we use dead cell removal kit from our partner, Multinei. And so as you can see in the bottom, at the bottom, we dramatically improved the quality of our cell suspension, and we completely flip the matrix. So we have over 70% of the live cells in both cases. There is no more cell debris that we can see, and we still have some clumps, but it's really much less than what it was before. So with this quality, we thought we were in good shape to proceed with our single cell experiment. In the lab, so other teams in R&D are looking a bit more in details in terms of how sample preparation can affect the downstream library that we are producing. So R&D at 10x collaborated with Multinei, and they are doing some experiments, and I'm showing you one of them, where they took 3 different tumors, so melanoma, colon, and breast tumors, and they proceed with only filtering, dissociated the tumor, and proceeded only with filtering only, or by adding filtering on top of filtering, RBC red blood cells lysis, and dead cell removal. And as you can see, by just looking at the library complexity and cleanliness, it dramatically improved the matrix in terms of the median genes per cell and the fraction of reads in cells. It also, when you clean up your cell suspension, it also improves the way you can count, your ability to count the cells in your cell suspension, and your ability to target the library that you want in your library. So those two metrics, the cells recovered in the library, are much closer to the targeted cell number than it was previously. We looked also at the reproducibility and the heterogeneity. So by just looking at the t-SNE plot in our Loupe cell browser, to see if the cell representation was not affected in the samples. There were a small improvement. We just noticed that in one of the tumor, we lost, with filtered only, which is a bit surprising, we lost completely one population. But this brings an important point, is that there is a lot of things that can be done to prepare those cell samples. And whatever we are doing, we need to be careful to not adversely modify the cell representation in our sample. So we just want to make sure that what we are studying is not removed during the cell preparation, and that you keep the cells as they are in the tumor itself. And I want to finish this section by just pointing you to our resources online in our support website. So we keep on adding a lot of demonstrated protocol and literature overall on our website. So I encourage you to come often to our website. And if in case you feel that we are missing a protocol that you would like on our website, or that you have a protocol that you really want that is very useful to a lot of people and around you, and you want it on our website, feel free to talk to our sales representative or call our tech support line. And with this, now that we have a nice cell suspension, I will give the tube to Sarah.
Thanks, Stefan. I'm Sarah, and I run the immunology collaborations here at 10x. I'm going to talk you through library prep protocol for generating both gene expression and VDJ libraries, so B cells and TCR libraries. I'll show you some data we've generated using the workflow, and I'm going to focus on two samples that we've looked at. One is a colorectal cancer sample, and the other is a non-small cell lung carcinoma. And then, as mentioned at the beginning, I'll do a brief demo using our Loupe cell browser and Loupe VDJ browser to show you the sort of interaction we can have with the data. The Chromium single-cell immune profiling solution is a complete end-to-end workflow. So you'd start with your cells that you've just generated your nice suspension from, and load those onto the Chromium controller. The complete solution includes all of the reagents and then the downstream analysis and visualization software. So here's a brief introduction to 10x technology. The Chromium, which is our box, is a droplet-based partitioning system. The input to this are the 10x barcoded gel beads, and these gel beads each contain the Illumina and oligos, which contain the Illumina read 1 sequence, a 10x cell barcode, a UMI, and a switch oligo, which is required for the reverse transcription reaction. The gel beads are passed through the partitioning device, as you can see in the video at the top here, and are mixed with cells and enzyme, and then partitioned into droplets by the addition of oil. So each droplet, or gem as we call them, acts as a chamber that contains a gel bead, the reverse transcription reagents, and 0 or 1 cells. And it contains a barcode that's on the gel bead that uniquely addresses that droplet and therefore that cell in the gem. So after partitioning, we collect the gem and perform the RT reaction and generate first strand barcoded cDNA with the barcode at the 5' end of the transcript. And then we remove the oil, break the gems, and clean up the cDNA. So once we have our barcoded cDNA, we can go forward with our library prep protocol. So the first step in the single-cell immune profiling solution after the RT reaction is amplification of the cDNA. So we amplify all cDNA using unbiased primers, and that's to generate a large pot of it that we can then use for multiple library preparations. So we take our tube of cDNA and we can take 1 aliquot of that and go forward with a gene expression library preparation, which is a standard library prep workflow: enzymatic fragmentation, endopair A-tailing and ligation, followed by a cleanup, and then the incorporation of Illumina indexes for sequencing. And we can take additional aliquots and go into our target enrichment protocol, which specifically enriches the TCRs and the IG sequences. So the enrichment is a two-step PCR, two PCR reactions, whereby the first PCR you prime the primers in the constant region of the TCR or the IGs. And then we do a second round of TCR with a nested primer to really give us the specificity that we're looking for when we want to see these transcripts. After those TCRs, we go through a library prep workflow, so fragmentation, end repair, as before, and then incorporate indexes for sequencing. So it's important to point out here that because the gene expression and the TCR IG libraries come from the same cDNA, we can use the cell barcodes to link the information together after sequencing and analysis, so we can get the same, the information of gene expression and the TCRs from the same single cells. And so, as I mentioned, we did this for our CRC and our NSCLC tumors, and we followed the workflow exactly as is written in the protocol. The libraries we generate are compatible with all Illumina instruments. There are some caveats when using the NextSeq in terms of quality, so we recommend against that where possible. The gene expression sequencing configuration is 26 base pairs read 1, 98 base pairs read 2, and the 8 base pair Illumina index read. And we recommend targeting around 50,000 reads per cell. For the repertoire sequencing libraries, it's a paired end 150 base pair read configuration with the 8 base pair index read. And here we recommend 5,000 reads per cell. We also have analysis pipelines, so you can run your data through Cell Ranger 2.1, which can process data both from the gene expression libraries and the VDJ enriched libraries, just by specifying different parameters. And this generates, among other things, outputs for the Loupe cell browser and the Loupe VDJ browser. I'm not going to go into any more detail on the analysis pipelines here because there are lots of resources available on our website that have been written by the people that wrote the algorithms and the software. And so I recommend you check those out if you want to dig deeper into how the analysis pipelines work. Okay, so now I'm going to actually get into the data that we generated from the CRC and the NSCLC tumors. We ran these through our Cell Ranger pipelines and then visualized the data using the Loupe cell browser and the Loupe VDJ browser. The Loupe cell browser is used to visualize the gene expression data, and the VDJ browser is focused on the repertoire sequencing data. So when we load our gene expression data into Loupe cell browser, the first visualization you see is the t-SNE projection of the cells. And if you're not familiar with these, each individual dot is a cell, and the cells are grouped together based on similarity in the gene expression profiles. So cells that are closer together in space are more similar. Here we classified the cells manually. So we looked at the top 50 genes present in each cluster, and then also looked at some known markers of specific cell types to make the call on what cell type was in that cluster. And you can see that by looking at these two different plots, we do see a huge range of different cell types, as Stefan highlighted at the beginning. We see epithelial, so tissue-specific and tumor cells, and these cluster together, sometimes really difficult to pull them apart into non-tumor and tumor-specific cells. And we also see a range of immune cells. So you see that there's a B cell population in both of these tumors, and there are T cell populations made up of subsets of T cells that we can identify looking at the gene expression markers. What's interesting here is that in the NSCLC tumor, we see this huge immune cell infiltrate making up more than 50% of the cells, and a very large fraction of it is B cells. So next we looked at the repertoire sequencing data for these tumors, for both the B cells and the T cells, and these are images output from the Loupe VDJ browser. And so just quickly go through what you're looking at here. You have the CRC B cell clonotypes, and these are histograms of the top 100 clonotypes, showing the proportion of the total clonotypes that each specific one contributes. So we have the CRC B cells, the CRC T cells, the NSCLC B cells, and the NSCLC T cells. And you can see that there are variable levels of clonal expansion in the different samples and in the different cell types. So interestingly here, what you see is despite the immune cell infiltrate being higher in the NSCLC tumor, there doesn't appear to be much expansion of either the B cells or the T cells. Whereas in the CRC tumor, we see that there appears to be a clonal expansion of a B cell, which is surprising perhaps, given that the infiltrate was greater in the NSCLC. So the really impactful thing about having these two different data sets for the same cells is that when you intersect them, you get this level of information. And you can link the gene expression transcripts to those that came from the B and T cells. And as this highlights, you really need both the gene expression and the repertoire sequencing data to understand what the immune infiltrate is actually doing, because it doesn't necessarily follow that just because you see a large immune cell infiltrate that there's any clonal expansion. So if I look a little bit more closely at that top B cell clonotype by intersecting the data set, so here I'm looking at an output from the Loupe cell browser, and I'll show you how to generate these plots shortly. So here, all the light blue dots represent any B cell clonotype. We'll just overlay those onto the t-SNE plot. And the dark blue dots you see in the top here are the dominant clonally expanded IG clonotype. And so if you remember from the initial t-SNE plot, this cluster at the top here represented plasma cells. So now we know that this top B cell clonotype is a plasma cell and likely producing antibodies, possibly against the tumor. You can take a bit more of a deep dive into this and look at the plot in the VDJ browser and also get the full gene segments and sequences for this clone. So I showed you these outputs from the Loupe cell and VDJ browsers. Now I'm going to briefly talk you through how we generated these. So I'm just opening up Loupe cell browser. And so Loupe cell browser and Loupe VDJ browser, you can download from our website, and you can also download example data sets. And here I downloaded the non-small cell lung cancer data set that I've just discussed. It's available on the website. So if I open up this file, the landing screen you come to is the t-SNE projection of the cells. And the initial view you see is a categories view and type of clustering. So here we're looking at graph-based clustering, and you can toggle this to k-means clustering. You can also come down to the bottom here and look at the log-fold 2 changes in gene expression. Here's a heat map of what's going on in all the clusters, and you can toggle this to a gene table, which gives you the top 50 genes driving the specific cluster. You can output these gene tables to CSV file by clicking here. So the other interesting thing you can do with the data is if you know what genes you're looking for, you go back to the categories drop-down menu and click on the gene expression, and you can search for your favorite gene. So if I'm interested in T cells, I might search for CD3, and you can see very clearly where the T cell clusters within this population. If you want to go into a little bit more detail on these T cells, you can search for your cytotoxic T cells. So search for CD8 and see what fraction of those cells are CD8 positive. And you can do the same CD4. And here we'll also light up other immune cell populations that express CD4. And if you want to specifically look at a subtype of CD4, so say T regulatory cells, you can look at FOXP3 and see that we have a small cluster of Tregs down here in this T cell cluster. So in addition to looking at what genes these cells are expressing, we want to know the clonotype. So we can use Loupe cell browser to look at that. We go back to this drop-down menu and select VDJ clonotype, and we can load in the T cell data set. And so here, everything that has a dot has an associated paired TCR clonotype. And you can get some more information on these in the Loupe VDJ browser. But first, if I click on this clonotype, you'll see it lights up as I showed you in the image on the slide. And if I want to know exactly what these cells are, there are a few ways to do it, but for a quick overview, you can toggle between the gene expression and the VDJ clonotypes options. So if I hit gene expression, and I looked at this data before, so I know that if I look at the CD8 positive T cells and look at this area down here where that clonotype fell, there's a very nice overlay. So it looks like these are CD8 positive cytotoxic T cells. You would need to do a bit more digging to ensure that that is the conclusion you should draw, but just an outline of some of the things you can do with this software. So if I go into the Loupe VDJ browser, I've already opened this file. So this is the T cell, the VLoupe file output by Cell Ranger. And you can see here that the landing page is the histogram of the clonotype. So you can toggle this between abundance, clonotype abundance, CDR3 abundance. You can look at V gene usage and J gene usage. And you can toggle that between alpha and beta, or alpha alone, beta alone. We go back to clonotype abundance. You can select the number of clonotypes you want to look at. So if you're only interested in the top 10, you can just look at those. And you can toggle between frequency and the proportion of the total clonotypes within the population. So I want to look at the individual clonotype sequences in more detail. You can click on them. And that takes you to a contig view. So here the consensus, which is the second bar from the top, represents the best summary of all of the contigs below. And at the top is the reference sequence. The colored bars represent features or points where the consensus and the contigs differ from the reference. So you can take a deeper dive into these sequences. So look at the consensus, for example. You can look at go into the details, see that that's the start codon. And you can jump within this view to different regions of interest, so such as the CDR3. And here you can see that in the consensus, there's a deletion and a mismatch compared to the reference, which you'd expect to see around these VJ junctions due to the chewback that occurs during recombination. If you're interested in exporting the data, you can copy and paste out the CDR3 amino acid and nucleotide sequences. You can also find like CDR3. So that's about it for the demo. I'm going to go back to the slides. Just an overview of the things you'd need if you wanted to run the demo yourself. We'll put these slides online, so later if you want to have a play with the software and the data, you can. And more resources available on our website. As Stefan mentioned, there's a whole host of demonstrated protocols available. We have very thorough software documentation for Cell Ranger and the Loupe Cell browser and Loupe VDJ browser. And there are also videos on library prep and tutorials on how to use the software. So with that, I will hand it back to Shauna.
Thank you, Stefan and Sarah, for a great presentation. We're going to get started with our Q&A session. Please remember to select the Q&A box on the bottom of the screen to submit your questions, and we'll do our best to get through as many questions as possible. All right, let's get started. So the first question is, do you have any tips on how to reach 90% viability on primary cells isolated by sorting from mouse organs?
Well, I can answer. this is a difficult question because it will vary from organs to organs. So my best advice would be, I mean, every organs require this, to troubleshoot the cell preparation for single cell experiments. So there is no really one answer for all the different organs. So I think it's a lot of trial and error. I would suggest to spend a lot of time troubleshooting the cell suspension to manage those, to get to those 90%.
All right, thank you. The next question, do you have a recommended dead cell removal kit for cells after cell sorting for a small number of cells, about 10,000 to 20,000?
This is, so the kit from Miltenay that we've used requires a lot more cells. I think the minimum is around 5 million cells. So if you have 10,000 to 20,000, that wouldn't work. This is a difficult number because for every centrifugation that you're going to do, you're going to lose cells. So it depends on the number of dead cells that you have in your sample. But if you are sorting them, I would remove the dead cells in your sorting scheme before you get to your samples. And one suggestion that what we've done with sorted cells is we do one pull down, I mean, one centrifugation to get to pellet the cells at the bottom, and we remove the liquid from the tube, and we leave about 5 to 10 microliters in the tube. We resuspend the cells in there, and we just load. We've done some stem cells, intestinal stem cells, for example, and we've loaded blind on our Chromium, and it works very well. We got thousands of cells. So we hesitated to do more centrifugation because, as I said, every centrifugation you lose a lot of cells. So we would minimize the centrifugation and minimize to handle the cells and go through the library pretty quickly.
Okay, and the next question, is there a lower limit to the number of cells to be able to use dead cell removal kit and still get a good yield after passing through the kit?
So the Miltenay kit, I think their limit is again about 5 million cells. So it's a column system, so we have column issue in terms of dead volume. So if you are below that, you start to lose a lot of cells. We've done about 2 to 3 million cells, and we got enough cells to run, plenty of cells actually, to run a single cell experiment. If you are, again, if you are below 100,000 cells, there might be advantages to do slow speed centrifugation to not lose as many cells. But again, centrifugation is also, each step is losing cells, so it's becoming very difficult at some point.
Next question. Is there any risk of biasing the cell type when using the live dead cell removal kit? Are there any cell types more prone to flipping PTD or to the outside?
So my answer is yes, it's possible. So that's why I mentioned in the presentation that you have to be very careful. Every step can be a risk to introduce a bias. So I would recommend, when you troubleshoot a dissociation protocol, I would recommend to check your cell suspension at the end by, sorting or fact analysis or immunofluorescence to make sure that you are not introducing a bias and that you keep the fidelity of the sample to the initial tumor, for example, the initial tissue, for example.
Right. With regards to the library sequencing, what are the caveats with using NextSeq?
I think with NextSeq we see that the overall quality of the read isn't quite as good, and so then when we look at our assembly of our paired sequences, we get lower pairing rate. It's not to say you can't use the NextSeq, but we see better data from the other Illumina sequences.
All right, and the next question, actually there are quite a few questions related to this. For the data analysis, how were the dead cells determined? What markers did you use, or how did you identify them?
Yeah, it's a good question. So in those clusters of dead cells, what we see is very high expression of mitochondrial genes. And when we look at the lists of the top 50 genes, it's pretty much all mitochondrial genes, so things that start MT-something. And we think they're dead cells because obviously if the cells are going through apoptosis, you see upregulation of those mitochondrial genes.
Great, thanks. And the next question, for these experiments, where were the tumors sourced from?
Yeah, I can take that as well. The CRC tumor we got from Conversant Bio. So they're a sort of biological sample repository that you can buy samples from. The NSCLC tumor sample came from a collaboration. So we're working with some people at a local medical center, and they gave us that sample for our experiments.
All right, thanks. For dead cell removal, can you use a kit from stem cell technologies as opposed to a Molteni kit?
I don't see why not. We haven't tested it. It's something that we have in mind to test. We've talked to them, to stem cell technologies to test it, but there is no reason why not using it. So if it's in your lab, just feel free to use it. Yeah.
Great. And the next question, could you please share the genes you used to identify the cells?
Which cells? I guess so. We would look, it depends on what cell type you're looking at, but I would say a good place to start is by looking at the top 50 genes expressed or upregulated by a specific cluster. And then if you know what genes your cell type of interest should express, you can then use the gene expression function within Loupe cell browser and type in the gene as I demonstrated, and see if it localizes to a particular cluster. It's a bit of an iterative process. There's not a defined list of markers, but I think I approached it in a similar way as I would to if I was doing a sort, like what markers would I be using to classify different subpopulations of cells, and then you can apply that to your gene expression data. But then of course you have a whole host of other markers you wouldn't have thought at to validate your thinking on what a particular cluster is.
Okay, next question. I'm not sure I understand how the cell barcode is kept when the amplification of TCR or BCR makes use of a C region primer. Can you please expand on that?
Sure. So the cell barcode is actually at the other end of the transcript. So the cell barcode is at the 5' end, and the constant region primer is at the 3' end. So at the 5' end, we're priming off a known sequence that is outside of the cell barcode and the UMI. So that's going to be maintained within your final amplicon.
Great. How do you identify clonal expansion in Loupe VDJ browser? By looking at proportion, the y-axis and the x-axis?
So this would be different people define clonal expansions in different ways. I would say we typically look at something that's more than 1% to start thinking about it being a clonal expansion. And when you toggle to that proportion view, it's a percentage. So when I showed you, and I can skip back through the slide, maybe. Here, so this proportion view, this specific clone's up at around 4%. And so we think that's an expansion.
Have you ever seen a population of eosinophils in single-cell RNA-seq, or do you see underrepresentation of eosinophils and neutrophils in single-cell RNA samples compared to sample analyzed by FACS?
So we don't have the orthogonal analysis of those tumors so we didn't analyze in the FACS or sorting the cells of the eosino and which was neutrophils. When we look at the single cell data, we found a small population of what we call basophils, but that's all we could find. It's very little cells, so it might be a number game if they are extremely rare in those tumors. So I don't think we've identified eosino as far as I know.
Thank you. Next question. Do you need to enrich T cells in the single cell preparation for TCR and VDJ analysis? What is the role of enrichment in the single cell preparation versus the 2 PCR enrichment steps for targeted? And what's the most important variable? The total number of T cells available in the sample or frequency of T cells in the sample?
I can start with the first pass.
Yeah.
The first part.
Mm-hmm.
So do you need to do a T cell enrichment in your samples? So it depends on your question. So we've done both. The good news is you get exactly the top clonotype. You find them without enrichment. You find them again in the enrichment. It's the low frequency clonotype that changes a little bit on the tail end. So the question is, how many clonotypes do you want to identify? If you want to get all of them, it's a number game, so you probably need to do an enrichment to get only the T cells, because when you use the unbiased, unenriched population, everything depends on the percentage of your T cells in the whole sample that you are running. So I guess it depends on what question you're trying to answer and what is important to you. But in both cases, the same clonotypes are appearing with enrichment and without enrichment, the top clonotypes. And for the second, is there a second part?
Well, the second part I think was about the enrichment PCRs. So you need to do those to basically pull out the sequences you're looking for. The immune receptor genes express at fairly low levels. So if you just look at your gene expression data, you don't really see them. So that's why we do those PCRs. So to really pull out the sequences and then be able to sequence them and see them at much cleaner, higher resolution than you would within the gene expression data set. And I think the question about whether you need to do enrichment like a CD3-based pull down and the PCRs, Stefan answered in that it sort of depends on your question. And if you don't want to see any other type of cell, then it makes sense to do an enrichment of the cells and then just go through the workflow and profile their gene expression and their receptors. But if you're interested in the tumor and you want to know what other cells are there, then you might not want to specifically pull out just the immune cells.
And there's one other part of the question. What's the most important variable in all this, the total number of T cells available or the frequency of the T cells?
I'm not quite sure what the question is asking. You have to have if you have a very, very low number of T cells and you try and pull them out, then you're going to have very few cells to actually load onto the instrument. So that might not be the best approach.
So probably enrichment would be a better way in that case to make sure to have all the...
To enrich and have a lot of cells.
Okay, thanks. Does 10x provide a cell type encyclopedia that defines specific cell types?
No, we don't as yet, but that's not a bad idea. It's something we can think about. I think the difficulty with something like that is that people like to use different markers to look at their cells. I mean, there are some core ones, but everybody sort of has slightly different favorite panels of genes they want to look at to call their cell types. And with immune cells, we're constantly discovering new subtypes which have new markers associated with them.
So hopefully with the Human Cell Atlas, we'll have a more extensive list of genes that we can look at in the gene expression. So we are not doing it, but maybe the Human Cell Atlas will answer those questions more extensively than we could.
All right, thank you. Just during the data analysis for the top clonotype identified, what is the list of top expressed genes?
I don't know off the top of my head. I'd have to pull up that data.
Well, how would you do that? How would you do that in the software? If you look at your clonotype, the top expressed clonotype, and then get the list of top expressed genes for that?
You can do that. I think that probably needs a demo on its own. Okay. I don't think I can just explain that briefly, but there might already be some resources on the website to sort of guide you through that. If there's not, let us know, and we can get something put together and put up to help you with that.
Next question, do you know the IG pairing efficiency? Also, is the current PCR scheme able to identify IG subtypes?
For IGs, the pairing efficiency is 80 to 90%, I think, fairly high. And yes, we do identify subtypes.
Thank you. Have you performed any benchmarking testing or preliminary testing on dissociated tumor cells from Conversant, which they now sell?
Say again?
Sorry. Have you performed any benchmarking testing or preliminary testing on dissociated tumor cells from Conversant?
Conversant Bio. Well, actually, we've used lots of their samples. So most of our human samples are from them, and it works very well. And the advantage also with Conversant Bio is that they can give you a profile, facts analysis profile in addition to the samples. So that can give you some orthogonal information there. So they work very well. Yeah.
And the next question, would it be possible that the digestion step would likely break tumor cells more than other normal cells?
Break in terms of killing them, I guess. Yes, because they are notoriously fragile. So yes, this is a risk. So this is why it's a very challenging sample type to work with because of that. So that requires a lot of troubleshooting on just preparing those samples for sure.
Great. Next question, can we use frozen PBMCs for immune profiling? Will we get comparable results to fresh?
Yes, absolutely. You can use frozen PBMCs. You know, you want to check the viability when you thaw them out, but as long as they look happy and healthy, then yep, you should get very comparable results.
What kind of raw data files come out that you can export to do further data analysis?
So I was specifically referring to the gene matrix files. They're particularly useful for using with other bioinformatics tools. But it depends what you want to look at. The outputs, you get BAM files. If you want to visualize the read localization using something like IGV, you could. You get the VLoop and the CLoop files for the browsers. And there's also a web summary, so you can look at the metrics and the standard charts we output to show you quality of the sequencing and basic pairing metrics for the VBJ and things like that.
Okay. When you say pipetting technique helps with viability, what do you mean?
Be gentle with the cells.
Okay, next question. Can you do hybridoma single cell sequencing?
Yes, why not?
All right. Next question. What is the best way to reduce clusters clumps from primary tumor dissociation?
So it's a delicate balance between dissociation, which can involve EDTA to prevent the cells from clumping, medium with 10x, 10% LBS, sorry, and also filtering to remove those big clumps. So it's a delicate balance because it's always to keep in mind to keep the representation of the cell population. So you want to dissociate enough, but not kill too much the cells, and dissociate enough so you don't have huge clumps and potentially lose cells in the cell preparation. So it's not an easy task.
Great. Can you expand on how to preserve cell population representation and how to check for it?
In terms of troubleshooting the dissociation, so I mean, I would look at the literature to find what are the best protocols that are out there, or probably in your lab. And usually when we've worked with lots of scientists to solve a bit different cell sample preparation. And one of the common themes is to start with the end in terms of getting a nice cell suspension with high viability and not too much cell clumping. And for the cell representation, I would recommend going back to maybe plating the cells on the slide and do some immunofluorescence to get an idea of what is the cell representation, or just do fact analysis or fact sorting. Try to see if it's something that is different that when you do other type or common type of analysis of your sample, and on the tissue itself also.
All right, next question. Do you recommend this solution for other species like rat?
So the gene expression side of the workflow is species agnostic. However, the targeted enrichment is specific for human in this workflow. We are very hard at work on a mouse version of the kit, and you'll be hearing more from us about that fairly soon. We don't currently have any plans to support rat, but if you were just interested in gene expression, then you can certainly follow that side of the workflow.
Great. For clear cell renal cell carcinoma cell dissociation, do you pull different regions from the tumor? If so, how many? How do you choose them?
Pull different regions? Oh, different. No, it was just grinding the tumor, and there were no special information or part of the tumor. It was just the whole tumor dissociated, so and frozen actually. But I mean, it's possible to do. We are actually doing this in R&D by just cutting pieces of the tumor and just processing only part. I mean, part of the tumor. So it's possible.
Do you have suggestion for calculating clonality evenness?
So there is a metric that's output on the web summary for clonality estimation, and that would give you some idea about the clonality of your sample. But you can sort of tell by looking at it. If you see that you have a few clones that are taking up, 5%, 10%, whatever percent of the overall clonal frequency chart, then you know that you have some more largely expanded things. Whereas if you see everything is down at 0.1%, then you know that you have a very, very diverse sample.
Okay. Is there a relationship between the number of UMIs and the multiplet clumps of cells?
Yeah, it's about filtering the data. So it's always a bit hard, but if you see outliers of really high UMIs, that might tell you something about it's possible that the cells is not unique in that UMI count. So it's one way to remove them. I mean, you can also, if you see that your expression is covering different cell types, that might indicate also multiplets. But yeah, I would say the first filtering, outliers in UMI counts is one way to proceed. Yeah.
Great. Next question: can single-cell immune profiling kit be used for frozen samples at -80 FFP samples or methanol fixed samples?
So the input to the workflow is a single-cell suspension. So you can certainly use cells that have been frozen and thawed, but anything that's been fixed isn't suitable for this workflow.
Thank you. All right, we're coming up on the hour, and we have time for one more question. Do you have a specific protocol for single-cell suspension from mouse brain tumor? I tried a protocol with different collagenases, but I observed a lot of debris even after filtering with cell strainers.
So we have fetal for sure. For adults, I'm not sure. If it's not on our website, I haven't used it myself for brain, but I know there is fetal embryonal protocol on our website. I don't know for adult brain. It's something that might require a bit more. I know people have done also nuclear preparation. That might also be a solution, although we haven't tried it for VDJ specifically. So I don't know if there is any protocols out there that would answer that question. So that might be something we probably need to work on.
All right, thank you. And that about wraps it up for today. Thank you to our 10x-perts, Stefan and Sarah, and to our attendees for great questions. We'll be posting a recording of this session on the 10x community shortly. Unfortunately, we had a lot of questions and we weren't able to get to them all. But if you have a follow-up question or we didn't get to your question, we urge you to post it on the 10x community in our single cell forum at community.10xgenomics.com, and we can address the questions there. And finally, if you're attending AACR 2018, we invite you to join us for our exhibitor spotlight on April 15th and a second exhibitor spotlight co-sponsored with Miltenyi Biotech on April 16th. Thank you and have a great rest of your day.